Scientists work very hard sometimes to assemble data sets. You can imagine a geneticist finding a population isolate somewhere in the far reaches of Greenland, and the investigator has lost 3 fingers to frostbite and so forth. They want to be able to exploit their data set. And the issue is figuring out how quickly, especially if this is funded with public funds, this data can be made public, but also in a way that allows the scientists to benefit. And I'll come back to all of these things. We as a community have actually failed to find good ways to credit members of large teams in papers, something that high energy physics has solved, but that's a relatively small community compared to the life sciences. And we certainly haven't solved the problem of how to credit people who publish lots of raw data without an associated paper. And then there are a lot of disagreements, both real and I think pseudo-disagreements, about what stage the data is cleaned and it's responsible to go out. Now, some people quote the example of the genomics community that during the Human Genome Project developed the ethic of putting all of their raw data out at the end of every working day. Not every data set is quite as simple as linear sequencing data, so that might or might not be the example. But I remember when I was an NIMH director and I was trying to get people in the autism genetics community. There were 6 or 7 groups who all had now, in retrospect, nobody really quite understood the complexity, so in retrospect, laughably small samples. And they were all arguing that they couldn't share their data and do a meta-analysis because they had asked their phenotyping questions slightly differently. And that really, we have to figure out when disagreements about data cleaning are appropriate and when these are really inappropriate. Now, as I've already said, sharing data allows us to, and reagents, by the way, allows us to address important problems that are otherwise recalcitrant. Often, people will say that big science, big data projects eat up federal funds, and already, I don't know that it would be a data project as much as a tool building project. A lot of the chatter around President Obama's announcement of a BRAIN initiative is what will happen to my R01. And I think we have to think about this, but very often at least when data sets are generated or tools are generated, these empower small labs. And certainly there were these same concerns about the Human Genome Project, but I think there's been vast benefit to individual labs, both by the data availability and maybe even more so by the technology that was built and shared. And which doesn't mean that all big science and big data science is good science. We have to be sensible, and maybe one of the desiderata will be whether it's going to move entire fields ahead. So just to give you one example, I've already alluded to it in genetics, but it's what I live with and what I lived with as NIMH director. You have to organize at the scale needed to attack the problem. So for illnesses like schizophrenia or autism or bipolar disorder, these are current centers providing patient samples collaboratively and sharing data in meta-analyses. And just to give you some sense of, for those of you who don't do this every day, this is called the Manhattan plot, but it actually looks more, doesn't look anything like Manhattan, because what you're supposed to see are skyscrapers. So these are chromosomes 1 through 22. The sex chromosomes are in here, arrayed at the bottom, and this is a statistical measure, and here is genome-wide significance. And at what seemed at one time like a large number of subjects, 7,000, sort of about half patients, half controls, nothing is genome-wide significant. So only when people share data and begin to reach numbers for, again, this is one example, the genetics of a complex disorder. Here, when all of those centers I showed you are sharing either samples or data from meta-analysis, now with 30,000 patients and 30,000 controls, there are 72 genome-wide significant regions, and actually, as of today, there's more than 90. And what's interesting is when the reason there are 90 now is when this result was shown at an international meeting, groups that had been holding on to their small data sets realized that it no longer made sense and contributed the data to the meta-analysis. And now almost everyone, there are a few companies that have, large pharmas that still have some data sets. Almost everyone we know of who have collected enough patients have contributed to this meta-analysis. And clearly, this is a beneficial result. And under each of these peaks, there's fine mapping to do and so on. But there's a lot of work now that lots of labs and lots of companies can do, turning these GWAS peaks into actionable biological data.